Hi, I'm Meghana Kshirsagar, and today I'm going to present BindVAE, which is a Dirichlet variational autoencoder based approach for de novo motif discovery from accessible chromatin. This work was recently accepted to Genome Biology. It's joint work with Han Yuan from Calico Labs, Juan Lavista Ferres from my team at Microsoft AI for Health, and Christina Leslie from Memorial Sloan Kettering Cancer Center. All results and models and source code from this work are available at this GitHub repository. Accessible regions of chromatin are generally regions that are permissive for transcription factor binding and are therefore hotspots of regulation of gene expression. Conversely, inaccessible regions where the DNA is coiled up around nucleosomes are less likely to be active regulatory regions. Among the various assays that have been developed to study binding of transcription factor to DNA, assays such as DNA-seq, MNase-seq, ATAC-seq look at all accessible regions of the DNA rather than only focusing on specific regions that are bound by a single transcription factor. So a single assay here gives us an atlas of tens of thousands of accessible peaks in a given cell type. Here we show the type of signal that is produced by assays such as ATAC-seq, which consists of peaks and then some regions with peaks and then some flat regions. Generally, we expect that dozens of TFs occupy overlapping subsets of these peaks due to the presence of their cognate binding sites or those of cofactors. The DNA sequence underlying regions of open chromatin is therefore complex. A key problem in regulatory genomics is interpreting the TF binding sites in all chromatin accessible peaks, as well as the TF specific regulatory grammars that allow TFs to bind at different locations within the same peak and at different types of patterns across different peaks. In this work, we build a Dirichlet variational autoencoder based approach that can take chromatin accessible data such as the one shown here and disentangle it into TF specific information, and more importantly also learn TF specific binding patterns that capture the particular preferences of transcription factors for the input cell type. Here is a preview of some of the results produced by our approach. Given an input DNA sequence such as the one shown here, BindVAE produces a disentangled output showing the particular TFs whose binding sites are present within this DNA sequence. It can learn TF binding motifs that are cell type specific. So we learn different TF binding patterns for GM12878 versus those for A549. The plot on the right here shows a projection of all transcription factor binding motifs that are learned, patterns that are learned by the model. We see some that are very similar and there are some that are very different and cell type specific. We also learn cooperative binding motifs where pairs of transcription factors that cooperate to bind to DNA sequence, we learn composite patterns for those TFs, for those pairs of TFs. There are several challenges, such as the size of the peaks being much larger compared to the size of the binding sites. The binding sites themselves can also be located anywhere within the peak. There are several binding sites within a single peak, and they can be overlapping. There's a lot of heterogeneity in the binding patterns of a single TF. For instance, FOXA2 likes to bind to patterns which can be of either of these two types. There are many transition factors with similar binding preferences, such as paralogous TFs, ALX3, ALX4, for example. Now let's take a look at our approach. Our model, BindVAE, is trained on ataxic peaks from a single cell type, and each peak is represented as a k-mer count vector. The model takes the DNA sequence, input DNA sequence, and projects it into a latent space defined by transcription factors. The assumption we are making here is that each DNA sequence is generated by multiple topics where the topics correspond to binding signatures of transcription factors. More concretely, the encoder consists of a multilayer perceptron that takes X and outputs alpha, which is the Dirichlet, which is the concentration parameter of the Dirichlet distribution, from which the latent vector Z is sampled. Z can intuitively be thought of as a membership vector that represents the input peaks membership in each transcription factor component. The decoder generates X prime from the latent vector Z via the distribution p theta, which we assume is multinomial. The goal of the model is to reproduce X prime that is as close as possible to the input X. BindVAE optimizes the parameters of the encoder and decoder jointly by maximizing the evidence lower bound, or elbow, using stochastic gradient descent. The elbow is the variational lower bound on the marginal log likelihood of the model and consists of two terms: the reconstruction loss, which is the first term, and the KL divergence between the prior and the posterior, which is the second term. Since the deep neural network has stochastic layers, we cannot use the typical backpropagation algorithm, and hence we need to use implicit reparameterization gradients in order to backpropagate gradients through the stochastic layers. This involves rewriting the Dirichlet distribution as a composition of univariate gamma variables. In order to make the model more useful for downstream tasks, we show here how we interpret the latent space that is learned by the model. On the left-hand side are the latent factors, where there is one factor per latent dimension, and on the right-hand side here are the entities to which we want to map them, which are transcription factors or transcription factor pairs or other types of genomic signals such as genomic background, cleavage bias of the enzyme being used by the assay. What we do to map these is that we take in vitro data such as high throughput SELEX probes from transcription factor binding experiments. We take all the enriched probes from a single transcription factor experiment and we take these probes from all TF experiments and we rank the probes for a particular latent factor and then assign that latent factor to the TF for which the probes are the enriched are ranked the highest. This gives us a mapping between the latent factors and transcription factors. Once we have this, we can use it to evaluate the model on various tasks. Coming to the results. In order to show that BindVAE learns diverse k-mer distributions, we here project probes from in vitro data from high throughput SELEX experiments into the latent space that was obtained by training our model on GM12878 ataxic peaks. And we visualize the resulting latent space, projections produced for the in vitro data. So note that while the model was trained on much longer sequences, DNA sequences of length 200 base pairs, here we are only projecting probes which are 20 base pair long into the same latent space. And Each row of this matrix here corresponds to one probe from one transcription factor experiment. The experiment is indicated by the label here. The first few rows here correspond to CPEB1. And there are 200 enriched probes coming from each ST-select experiment. So note that the probes thus represent DNA sequences that have a very high uh, binding propensity uh, to the transcription factor of interest. We see this nice block diagonal like structure here in this visualization, which shows that individual components, individual latent factors learned by our model are learning the binding preferences for different and separate transcription factors on the one hand, whereas on the other hand we see some latent factors capturing binding preferences for families of transcription factors. For instance, here all fork head transcription factors seem to lie in the same latent space given by these two components 96 and 97. We find that BindVAE learns k-mer patterns that correspond to binding patterns of transcription factors as verified by comparing to motifs that we see on cis-BP. For instance, here we show top 50 k-mers from the k-mer distributions learned for NFKB1 and TFAB4. We find that some k-mers capture the prefix of the motif and some others capture the suffix of the motif. We further find that BindVAE disentangles the binding signals in input DNA sequences, where we see that for this DNA sequence, two TFs are found. Here is another peak which only contains repeat regions, for which only the latent factor corresponding to low complexity patterns is enriched. And here is another peak which has both repeat regions as well as TF binding signal. We further find that BindVAE learns different TF binding patterns for the same TFs for GM12878 and A549. So these are patterns learned by the two models trained on these two separate on peaks from these two cell types. In order to obtain composite motifs from BindVAE, we project CapSelect's probes into the latent space learned on ataxic peaks, and we find latent factors that enrich these probes. We then visualize the k-mer distributions of these latent factors and compute the motifs corresponding to the k-mers of those latent factors. As you can see, we find FOXJ3 TBX21 amongst one of the cooperative binding motifs that we find. We also compare with de novo motif discovery approaches, in particular such as Homer, and we find that out of all the expressed genes for GM12878, BindVAE finds more transcription factor binding signatures as compared to Homer, which it has a higher precision and recall. Additional results showing all the motifs learned by BindVAE, evaluation on more cell types, performance on ChIP-seq, and so on are in the paper. To wrap up, there is a lot of prior work on both de novo motif discovery approaches as well as chromatin feature transcription factor binding prediction. Our approach has the following key strengths over prior works. It's fast, unsupervised, it learns cell-dependent representations and requires only one DNA accessibility experiment. It can work with less studied cell types where TF-specific experiments such as ChIP-seq are lacking, and it's a very easily interpretable model. Further, we find that our model does better than prior de novo motif discovery approaches on the task of retrieving transcription factors that are expressed in a cell type. Here is a link to our full paper and the dataset, source code and models.